Now, having observed the cells using both the light microscope and the electron microscope, let us see how we can estimate the size of these cells. This can be done in the laboratory using a light microscope. Now, prior, a temporary slide will be prepared having the cells. Now, for convenience, plant tissues are much easier to prepare and use compared to animal tissues. So in the procedure, the first thing that is to be done is to determine the diameter of the field of view. Now the field of view is that lit. There's an area that appears as lit, the circular area. That is the circular area that you see when you look through the microscope. So the first thing that is done is you determine the diameter of this field of view by placing a transparent ruler marked in millimeters on the stage. Then you focus the microscope so that the millimeter markings on the ruler appear as dark lines. So once you place the ruler or even a set square with millimeter markings, then you focus, and this is how it's going to appear. The dark bars here represent the millimeter markings. Now position the ruler across the center of the field of view with one dark line of the millimeter. This one, for example, the dark line of the millimeter marking touching the left edge of the field of view. This will make it easier to determine the distance across the diameter. So you place the ruler such that one of the markings touches the left edge of the field of view. Now remember that 1 millimeter is the distance, this distance here between one marking and the next. That is 1 millimeter, second millimeter, third millimeter. So you determine the diameter of the field of view by counting the number of spaces between the millimeter markings. In this particular case, there are 3 spaces, so which means the diameter of the field of view is 3 millimeters. Next step, you convert the diameter of the field of view into micrometers. Remember, millimeters are too large to use in microscopy. The cells are tiny, they're microscopic, so we must use units that are convenient. Otherwise we'll be dealing with so many decimal places. We don't want that, so you convert it to the micrometers, and 1 millimeter carries 1,000 micrometers. So you've determined that the diameter of a field of view here is 3 millimeters, so you convert that by multiplying by 1,000, and that gives us 3,000 micrometers. So the unit of micrometer is this letter mu, then m. So that stands for micrometer. So now we know that our field of view has a diameter of 3,000 micrometers. Then next step, we count the number of cells along the diameter of the field of view. Now to do that, you'll have to remove the ruler and place the microscope slide with the cells on the stage. You then position the slide so that a row of cells are arranged lengthwise like that. Of course, there are many other cells, but you focus on a particular row and position them in such a way that they run across the diameter of the field of view. Then you simply count the number of cells in the row. In this particular case, you have 1, 2, 3, 4, 5, 6. So we have 6 cells covering the entire diameter of the field of view, which has a dimension of 3000 micrometers. So the next step is just calculating or by estimating the length of an individual cell using the formula. The length of one cell is calculated by dividing the diameter of the field of view in micrometers by the number of cells across the field of view. So in our particular case, we determine the diameter to be 3000 micrometers, and that this distance was covered by 6 cells. So divide by 6. This gives us an average length of one cell to be 5000 micrometers.